Hello, myself Gautam from 316. Today my topic is biological oxidation. Biological oxidation is the process where cells break down and nutrients like glucose to release energy. This happens through a series of reactions where electrons are transferred from the nutrients to electron receptors, acceptors, usually oxygen. As electrons move through these reactions, energy is released and stored as an ATP, the cell's main energy source. Biological oxidation primarily takes place in the mitochondria and involves pathways like glycolysis, the Krebs cycle, and the electron transport chain. This process is essential for energy production in cells, supporting growth, repair, and other vital functions. Here's a breakdown of the process. First one is electron transfer. In biological oxidation, electrons are transferred from organic molecules like glucose, fats, or proteins to electron receptors through oxidation reduction reactions. When a molecule loses electrons, it is oxidized, and when a molecule gains electrons, it is reduced. Second one is energy release. During oxidation, energy stored in the chemical bonds of the organic molecules is released. This energy is harnessed in the form of adenosine triphosphate, the primary energy currency of the cell. Third one is enzymes and coenzymes. Biological oxidation reactions are catalyzed by enzymes which accelerate these reactions under biological conditions. Coenzymes like NAD plus, FAD plays a key role by acting as electron carriers, shuttling electrons through various stages of metabolism. Fourth one is aerobic and anaerobic oxidation. Aerobic oxidation. When oxygen is the final electron acceptor and the process it is known as aerobic oxidation. It is highly efficient and occurs in the mitochondria of eukaryotic cells, generating a large amount of ATP. Second one is anaerobic transport chain. In anaerobic organisms, the electrons from NADH and FADH are passed down the electron transport chain in the inner mitochondrial membrane, where this energy is used to pump protons across the membrane, creating a gradient. This gradient powers ATP synthesis in a process called phosphorylation. This biological oxidation after part of a cellular respiration. It involves a series of biochemical reactions that cells use to extract energy from nutrients like glucose. The main steps are as follows. First one is glycolysis. It occurs in the cytoplasm, breaks down one glucose molecule into two molecules of pyruvate, produces a small amount of ATP and NADH. Second one is pyruvate oxidation. Takes place in the mitochondrial matrix, converts each pyruvate molecule into acetyl coenzyme, produces NADH and releases carbon dioxide. Third one is citric acid cycle, also known as Krebs cycle. It occurs in the mitochondrial matrix, acetyl coenzyme. Third one is citric acid cycle, also known as Krebs cycle. It occurs in the mitochondrial matrix. Acetyl coenzyme enters the cycle and undergoes a series of reactions, produces ATP, NADH, FADH, and releases CO2 as a waste product. Fourth one is electron transport chain and non-oxidative phosphorylation. It takes place in the inner mitochondrial membrane. NADH and FADH donates electrons to the electron transport chain. Electrons move through protein complexes, creating a proton gradient. The flow of protons backs into the mitochondrial matrix through ATP synthase and drives the ATP production. Oxygen is the final electron acceptor, forming water as an byproduct. Fifth one is ATP synthesis. The proton gradient generated by electron transport chain powers ATP synthesis to produce ATP. This step is the main source of ATP formation in anaerobic respiration. Overall, biological oxidation is a key component for cellular respiration, allowing cells to obtain energy from nutrients and maintain metabolic functions essential for life. Thank you.